Great. So our next speaker is Minna Roh-Johnson, and she will speak about the dynamics of innate immunity-guided tumor cell motility in vivo.
Well, thank you first to the organizers for this great opportunity. Over the last 10 years, I've been fascinated with the regulation of cell migration in vivo. And so today I'll be telling you a story about how stromal cells in the tumor microenvironment affect melanoma metastasis. So my work in, It doesn't appear to be slide advancing. Here we go. My work in cancer cell biology first began in mouse models of human breast cancer, where we transplanted in human cells, shown here in green, into mice where the vasculature was labeled in red and collagen fibers in blue. And what we were interested in is what components in the microenvironment affect these cells' ability to migrate versus non-migratory counterparts. And from a great deal of work done by many different labs, we know a little bit about how this process occurs. So shown here in this schematic, there are tumor cells in blue. Tumor cells can break off and migrate through the stroma, and there they interact with components of the microenvironment, one of which being tumor-associated macrophages. Now, macrophages are part of the immune system, whose traditional role is to help the host. However, there's a subset of macrophages in the tumor microenvironment that are differentiated to be pro-tumorigenic. So they signal to tumor cells to promote invasion. They also signal with tumor cells to promote intravasation into the vasculature. And at this time, I became curious about whether these pro-tumorigenic functions of macrophages were true in other epithelial cancers, such as melanoma, and whether we could use additional model organisms to really tease apart the dynamics of this process. And for this reason, and what was beautifully illustrated in the previous talk with the high-resolution imaging of tumor cell motility, we started Zebrafish experiments. So the approach that we use is a xenotransplantation approach, where we put human melanoma cells in, but this is in the 2-day-old larvae prior to formation of the adaptive immune response, so the fish take up the human cells very readily. It also gives us a unique opportunity to just look at interactions with the innate immune response. Because we imaged the site of injection, we did not inject near the yolk, but instead injected into the hindbrain ventricle. And once in that ventricular space, the tumor cells integrate into the overlying skin. And that allows us two things. First, the cells are in the proper microenvironment of the skin. And secondly, it's close to the surface, allowing it for high-resolution imaging. So here it is at low magnification, a green tumor cell in the skin of the fish, and here at high resolution, where this human melanoma cell is expressing LifeAct, look at actin, and you can resolve subcellular structures as this tumor cell is associating with the zebrafish vasculature. Okay, so importantly, when we do transplant these melanoma cells in, they do disseminate. Here are two examples of dissemination. And we went through a panel of metastatic melanoma cell lines to show that they all disseminate at varying degrees. So each dot in this graph is an experiment comprising of anywhere between 20 and 40 larvae. So 40 means 40% of the fish in that clutch looked like something like this. This was true for human metastatic melanoma cells, but also for zebrafish melanoma cells. We acquired cell lines from Richard White. And importantly, when we put in non-metastatic cells, they do not disseminate to the same degree, suggesting that this dissemination is a feature that is specific to metastatic cells. But recall that I mentioned before that we were really interested in teasing apart the dynamics of tumor-associated macrophages with tumor cells, and we thought, first we would determine whether macrophages are pro-tumorigenic in our zebrafish system, and then if that was true, we could tease apart how these cells were signaling with one another. So the first thing we did is to look at the interactions between the tumor cells and the macrophages. The macrophages are in red. And we see close interactions between the tumor cells and macrophages. The macrophages are very dynamic. They extend out long, thin protrusions, and oftentimes the macrophages make prolonged contacts with tumor cells. We were concerned that this could be due because it was a cross-species interaction. So we looked at zebrafish melanoma cells, so this is now zebrafish tumor cells with zebrafish macrophages, and we saw similar dynamics, suggesting that it isn't due to a cross-species interaction. But then we wanted to determine how are these two cells talking to one another. So there were three strategies that we tested, one being secreted signaling molecules, like a ligand is sent out to a receptor in a target cell. Another was cell contact mediated signaling, so close apposition of those cells mediating signaling in the target cell. And the third one being cytoplasmic transfer of components, a relatively new way that's been discovered for cell communication, where the cytoplasm of the signaling cell is taken up by the target cell, either extracellularly by being packaged up in these tiny vesicles called exosomes, or by cell contact mediated measures where a long, thin, narrow tube called a nanotube, open-ended on both sides, acts as a conduit where the cytoplasm of one cell can move to the cytoplasm of the other cell. So for reasons for time, I'll just be talking about this third one, cytoplasmic transfer. The first thing we wanted to determine is there cytoplasmic transfer, as in, can macrophages transfer their cytoplasm to tumor cells? And the way that we looked at this is using an approach that was adapted from Jaco van Rienen's lab, where tumor cells were stably expressing this construct with DS-red that was floxed, followed by GFP. So all cell lines expressing this construct were red. However, if it received Cre, the red would be floxed out, and these tumor cells would turn from red to green. And we expressed Cre specifically in macrophages. So in a case where there's no cytoplasmic transfer, no Cre would get into the tumor, and the tumor would stay red. However, if there was cytoplasmic transfer, you would see cytoplasm as well as Cre getting into the tumor, and you'd see this color switch from red to green. So the presence of green tumor cells was an indicator for cytoplasmic transfer from macrophages. So the experimental approach looked something like this, where the zebrafish had Cre driven with a macrophage-specific promoter, and the tumor cells expressing this LOXP construct was transplanted in, and we asked, in that red clump of tumor cells, do we ever see green cells, indicating transfer from macrophages? Okay, these are the results. So here's a lateral view then of the fish, anterior, posterior. This red clump here is the transplanted tumor cells, which are DS-red. The red heart indicates the presence of the transgene. And when we look at the same fish in the GFP channel, you see a single GFP-positive cell right here, indicating transfer. And we quantify this as percent fish with green cells anywhere in the body. When there's no Cre, we don't see green cells because there's no floxing of DS-red. However, when we drive Cre specifically in macrophages, we now see the presence of green cells. So what this suggested to us is, yes, macrophages can transfer their cytoplasm to tumor cells. And what we next wondered is, what does this have to do with metastasis? So we performed the same experiment. Again, red tumor cells, no transfer, green tumor cells, transfer. And we asked now just looking at the disseminated cells, what color were they? Were they red or were they green? And this cell line that we use, recall the panel that I showed before. The cell line that I'm using here has a 14% dissemination rate. And what I'm graphing is just the percent green cells here. So here's the average 14% dissemination rate. What I'm graphing is just the disseminated green cells. In a control situation with no Cre, you would expect those disseminated cells to just stay red. That's in fact what we see. They almost all stay red. However, when we drive Cre in macrophages, now we see that for almost all of the dissemination we observe, almost all of them are green. Okay, so what this suggests to us is that macrophages can deliver cytoplasm to tumor cells and that they may confer that metastatic capability. To determine whether this was true specifically for macrophages, we also drove Cre in two other components in the microenvironment, saw that they looked like control, suggesting that this feature was specific to macrophages. Okay, so if this is true, if macrophages can transfer their information to tumor cells, and that gives the tumor cells ability to migrate, is this a cell contact mediated event? So to do that, we hypothesize that if we increase or decrease cell contacts, we should increase or decrease dissemination. So that's what's shown here. Tumor cells, again, are in green, macrophages in red. We did a reverse genetic CRISPR-Cas9F0 screen of candidate genes and identified MyD88, a component of the inflammatory response pathway. This is now in the mutant, and in the mutant you see reduced macrophage infiltrate. And how did that affect dissemination? Well, we also saw a reduction in dissemination. So then we asked if the converse is also true. If we increase tumor cell macrophage interactions, do we see an increase in dissemination? So the way that we did that is we created a constitutively active point mutation in MyD88 and then drove it specifically in macrophages. And you can see this increased infiltrate of macrophages in the microenvironment as well as a concomitant increase in vivo dissemination. So what this suggested to us is that macrophages can impact the tumor cell's ability to disseminate in a cell contact mediated manner. And then we asked, is this cell contact also required for this cytoplasmic transfer of material that we observe? So to do that, we try to capture the events as they occur in real time. So this is experiments that are ongoing, and I'll show you a movie of an example that we have. This is imaging at the site of injection, where the tumor cells are expressing, again, this LOX-P construct. So here they are in DS red. The macrophages are also in red, but they're caxed, so you can see, and they also morphologically look different, so we can always tell the difference between macrophages and tumor cells. Nevertheless, you can see that tumor cells and macrophages are in contact with one another, and when you look in the GFP channel, it is not yet GFP. And when I play this movie, what you'll see is macrophages and tumor cells maintaining contact with one another as a tumor cell begins to turn green. These bright green irregular balls are debris that the macrophages have eaten up. So I'll play the movie. You can see that if you look in this channel first, a little loop, the tumor cell maintains its contact with macrophages for about 4 hours. And you'll see, again, when this loops, the tumor cell slowly starts to turn green. So what this suggests is that cytoplasmic transfer of components from macrophages to tumor cells is a cell contact mediated manner, and from our other experiments that this cytoplasmic transfer regulates dissemination. So here's our working model. In the primary tumor, there's a subset of tumor cells that can interact with components of the microenvironment, one of which being tumor-associated macrophages. The macrophages can then deliver their cytoplasm to tumor cells, either by exosomes. However, we think that exosomes are systemic, and the results that we have suggest that it's cell contact mediated. So we favor a hypothesis where instead the cytoplasm is delivered through nanotubes, allowing for the free flow of cytoplasm from one cell to the other. And indeed, Paul Martin's lab had shown in the early larvae that immune cells and neoplastic cells form these long, thin tethers that do resemble nanotubes. Regardless of the entry of cytoplasm to the tumor cells, we think that this cytoplasmic transfer is what contributes to tumor cell metastasis. And indeed, in some of our movies, we can see tumor cells extending out protrusions toward macrophages and this tumor cell's ability to translocate from the primary tumor. So we are now commencing mouse experiments to ask whether we can observe this macrophage-dependent cytoplasmic transfer in mammalian systems. We're also doing in vitro approaches to really nail down whether this transfer occurs through exosomes or nanotubes, because we can resolve those structures in vitro. But really importantly to us, we want to know what is being transferred, what are the biomolecules moving from macrophages to tumor cells, and we are planning some profiling experiments to address that. So with that, I'd like to thank the entire Moens lab, present and former. It's been a wonderful environment to work, especially Arish Shah, a technician who did almost all the cloning for this project, as well as our Zebrafish technician, Rachel Garcia, who's been really helpful for everything, especially screening for founders. John Condilis, my former postdoc advisor where I did the breast cancer studies, and the following very generous people for reagents and advice. Thank you.
So I have a question. Do you know if the transfer is bidirectional, and have you imaged more the macrophages that have contacted extensively a tumor to see if they were reprogrammed to keep doing
Yes, yeah. So from the literature, we know that the transfer can be bidirectional, in nanotubes specifically, actually exosomes also. However, I don't know in our system because I haven't done the reciprocal where the Cree is in the other cell in them, and that would be very interesting to know. With respect to how would I know if the same macrophage keeps hitting it, so there is a dendra that's under the control of the macrophage-specific promoter that I've dabbled with a little bit, where once they touch, we could photoconvert them, thereby marking them, and then ask if the same one hangs out repeatedly. And it would be really interesting to know that. Thank you. Yeah.
So MyD88 is also sort of intimately involved in the activation state of the macrophage, and I'm wondering how you uncouple the cell-to-cell contact with the activation state.
Yeah, that's a really good question. So I don't think with the MyD88 experiments that we can uncouple specifically cell contact versus just recruitment. So really, I think we're affecting recruitment, right? The infiltrate into the tumor. And by proxy, I could say, well, if they're not near the tumor, they probably aren't making physical contacts with tumor cells. But I tried to think of a way where I don't really affect recruitment. I only affect cell contact. But that would require usually drugs of actin regulators, which would destroy motility altogether. So I just haven't found a very good way. But if anybody has any suggestions, I would love to know.
I'll talk to you later.
Yeah, thanks.
It's a beautiful story. I think I missed something. So if you have a red tumor and a Cree macrophage, and then some hours later you see a green spot elsewhere in the embryo. Can you tell if that's a tumor cell or a macrophage? And do you also see, other red cells around it? Or is it always the same? How does that work out?
Yeah, okay, so the first question, how do I know that it's a tumor cell versus a tumor cell that got eaten by a macrophage, say?
Or maybe there is that other transfer.
Right. So we did look at the disseminated, so what happens at the primary tumor, the movie was just at the primary tumor where you see the color switch. I then have separate sets of experiments where I see disseminated cells. What I haven't been able to do is link it all in a single movie where it changes color and then I keep it and all of a sudden I see a green cell appear and it's the same green cell that converted. I at least know that it's not being eaten by macrophages because I can look at the disseminated cell and ask if a macrophage ate it. The other thing is you can tell by size because then it's a irregularly shaped ball that's much, much smaller. The second question, I'm sorry, I don't quite recall.
I think I got the second one now.
Okay, thank you.
Thank you very much.